Good morning to everyone. This is Wednesday morning. It's 10 a.m. Houston time, so I have to say hello to all the large family of MD Andersonians from inside the institution, from medical center, and from abroad in US and all the world. Thank you all the attendees. We know how stressful the life is these days, so thank you very much for being with us, because without you, the one who are listening and asking questions, this type of webinars certainly would not make sense. And in fact, we are very much happy for the speakers who are presenting, and today we have a wonderful speaker, Dr. Michael Davis, and wonderful topics. I have to say thank you to the people who organize this Enjoy Science, and there are under Giulio Draeta office, and thank you, Giulio, for organizing this one. And of course, thank you to Liz Kent and to Ellison Garth, because they are basically the basis who are keeping this running and keeping this organizing. And it's my pleasure to host Enjoy Science with my friend and colleague Alessandra Ferraioli. And with this said, let's welcome Dr. Michael Davis. Everyone from MD Anderson family know Michael, because he's one of the most collegial person in this institution. And of course, I have to tell you that what things that you know, he's the chair of the Department of Melanoma. I don't know if you know that he was named chair at one of the youngest age in the history of the institution. Why? Because in this country, hard work and good jobs are paying back. And in fact, Michael was wonderfully trained in Massachusetts General Hospital. Then he came back about 15 years ago in MD Anderson and built his career from fellow through becoming a chair. And he has extraordinary achievements, wonderful publication and wonderful clinical activity. He has done a lot of research, from basic research, genomics, till clinical translational. And why I'm prizing Michael, it's because being a chair, being so busy, I have to tell you that I was listening one of his presentations for the postdocs, an educational presentation that was one of the most beautiful that I heard. So Michael is not only an extraordinary clinician, an extraordinary scientist, but he is a wonderful mentor. And in fact, it's excellent that we have in our institution such clinician scientists who pay so much time and so much patience for mentoring young people. And with this said, let's give the show to Michael. Thank you, Michael, for being with us today. And we are looking very curious for your presentation, because the title, it's very interesting. Good luck with everything, and thank you again for being with us.
George, thank you so much for that introduction. That's very, very kind of you. And actually, just as adding a little bit to that, for those of you who may have known me for a long time, I did my first rotation in a laboratory here at MD Anderson in the summer of 1994, and I did the PhD component of my MD PhD training on primary brain tumors and glioblastoma with Peter Steck, who had discovered the P10 tumor suppressor gene. And the interesting part of my career is that, having started working on primary brain tumors, really the focus of my research has evolved to brain metastasis. And it's really exciting to be able to talk to you about not only the work that we have done, but the progress that we've made, not only across this field, but particularly for patients. So I really appreciate the invitation from Dr. Kayla, Dr. Ferrajoli, to talk to you about this research today. Very briefly, these are my disclosures. As George mentioned, as a physician scientist, we're very focused on trying to translate our work into new clinical trials. And so we tend to work with a lot of different companies about trying to find strategies that we can end up taking to patients. And I also am involved in clinical trials. So just as a bit of brief background to talk about the significance of brain metastases have always been the thing that patients fear. And, over the last decade, we've seen tremendous progress in so many new, more effective treatments for patients with cancer. And one of the interesting sort of consequences of this is that as we see these treatments improving the survival of patients with metastatic cancer, it looks like we're starting to see more and more patients developing CNS metastases as they live longer and longer. And so it's estimated that each year between 98,000 to 170,000 patients are diagnosed with brain metastases. This represents approximately 10% of all cancer patients and up to 40% of patients with metastatic cancer. And again, these are very significant and often lethal events, and it's estimated that brain metastases cause approximately 100,000 deaths per year from cancer and up to 50% of patients that are dying from cancer have brain metastases. As shown to the right, the most common sources of brain metastasis are lung cancer, breast cancer, which are again very common malignancies, but actually the third most common source of brain metastases is melanoma, which I'll talk about a little bit more. So we know that there are certain cancers that have a high risk of brain metastasis. Interestingly, other cancers like colorectal cancer that have a much lower rate of metastasizing to the brain. So again, an increasingly common problem and one for which better treatments are needed. And one of the challenges that we face as clinicians is the fact that patients with brain metastases have historically been excluded from the overwhelming majority of clinical trials. And so this is data from a paper by Dr. Nancy Lin at the Dana-Farber Cancer Institute. She published a couple of years ago reviewing almost 1,500 clinical trials designed for patients with metastatic breast cancer, and showed that not only were only 2% of those trials designed for patients with brain metastases, almost 50% of trials excluded patients with any history of CNS metastasis, not just with having current CNS metastases. And so again, one of the big challenges that we fight in the clinic is simply increasing access to patients for new agents. And while historically this has been partly justified due to the poor outcomes of these patients, that's really starting to change. And so focusing on those most common cancers, talking about first lung cancer, we've seen over the last several years multiple targeted therapies addressing alterations in EGFR and ALK that have shown very impressive activity in patients with brain metastases. And these include agents that have been designed specifically to cross the blood-brain barrier, where in patients who've developed brain metastases and are now in the second-line setting for targeted therapies, these agents are not only effective, but they're often showing greater efficacy against brain metastases than against extracranial tumors. And here at MD Anderson, we're really very excited and proud about the work of Dr. Roshni Murthy, who led the HER2 CLIMB study for patients with HER2-positive metastatic breast cancer. And this was one of the first randomized trials that allowed patients with untreated or progressing brain metastases, who ended up constituting almost half of the patients in the trial. And tucatinib ended up getting FDA approval because at the addition of tucatinib improved progression-free survival and overall survival in the whole cohort. As you can see in the lower right, this is one of those trials where we actually saw more benefit in patients with brain metastases than in patients without brain metastases. And so it was really data like this that's really been challenging the dogma of excluding patients from brain metastases from clinical trials. And I was really very excited and fortunate, along with several other colleagues here at MD Anderson, to participate in 2019 in a workshop that was organized by the FDA, specifically talking about product development for CNS metastases. And here are two of the real takeaway messages. First, were the fact that companies and investigators will need to provide clear justification in order to exclude patients from with brain metastases from early phase clinical trials. Again, trying to change what was the status quo, reflecting not only the new data, but this clear unmet need that we have moving forward. And really, I think the second consensus that came out of this meeting was the fact that there is a continued need for better understanding and models of brain metastasis in order to develop more effective therapies. And so with that as background, I'd like to spend the rest of this time then talking about the work that we've been doing in melanoma to understand and improve treatments for patients with CNS metastasis. I'll talk first about where we stand with systemic therapy, including recent trials with both immunotherapy and with targeted therapy, and then talk about the insights that we've made into the pathogenesis and resistance of these tumors and what the therapeutic implications, and finally a brief talk about some of our future challenges, but also opportunities in this space. So for those of you who aren't familiar with melanoma, fortunately melanoma is not the most common skin cancer. That's really squamous cell carcinoma and basal cell carcinoma. And melanoma really only represents about 5% of the skin cancers that are diagnosed each year, but melanoma is by far the most aggressive of the common forms of skin cancer, and it's responsible for approximately 80% of skin cancer-related deaths. One of the things that really stands out about melanoma is its proclivity to be able to metastasize to any organ, but particularly to spread to the CNS. So although breast cancer and lung cancer are the most common source of brain metastases numerically, The fact that melanoma is the third most common source of brain metastasis while being a much less common malignancy reflects the fact that melanoma has really the highest risk of our common solid tumors to metastasize to the brain. And indeed, one of the striking features is that up to 20% of our patients show CNS involvement at the diagnosis of metastatic disease. And in historical series, we see up to 50% of patients develop clinical evidence of brain involvement over the course of treatment. And in autopsy studies, over 80% of patients had CNS involvement. And this, again, has been a common site of treatment failure, particularly for chemotherapies, biochemotherapies, and targeted therapies. Now, in the old era of melanoma treatments, the median survival for patients diagnosed with CNS metastasis from melanoma was about 4 months. And research that we had done had demonstrated that by far the group with the worst outcomes are patients with leptomeningeal disease, which means tumors that are now on the lining of the central nervous system, either in the brain or the spinal cord, something that I'll come back to at the end of this talk. And again, we started out as a disease treated with chemotherapy, including blood-brain barrier penetrating chemotherapies, which would really achieve significant tumor shrinkage in patients with brain metastases only about 5% of the time. Now, as I mentioned, that's really sort of old history in melanoma, and over the last decade, we've had 12 new agents approved for patients with distant metastatic disease. And these are really evenly divided between immune therapies and then targeted therapies for patients with the BRAF mutation that's present in about 50% of patients. Earlier this year, we had our first immunotherapy targeted therapy combination regimen receive FDA approval with vemurafenib, cobimetinib, and atezolizumab. Now, thousands and thousands of patients were enrolled into the trials that led to the approvals of all of these different agents, and yet not a single one of those registration trials included patients with active brain metastases. They were excluded from all of those studies. So again, very similar to what we were describing was also the historical nature of what we had seen in breast cancer. However, what we've done is since these agents were approved, is several of the agents have been evaluated for safety and efficacy in post-approval studies in patients with CNS involvement. So what are some of the lessons that we've learned from those? So again, immunotherapy has really transformed this disease and the outcomes of patients. And the first significant trial we had for immunotherapy in patients with active brain metastases was ipilimumab, of course, developed by our own Jim Allison, and the first checkpoint inhibitor to be approved for patients with metastatic melanoma, a treatment that has a response rate in patients without CNS metastasis of about 15%. This trial included two groups of patients. Cohort A were patients with asymptomatic brain mets not requiring steroids, and we can see that in those patients, the response rate in the brain was 18%, very similar to what we see in patients without CNS disease. They also included a small cohort of patients whose tumors had enough edema or symptoms that the patients required steroids, and here we actually saw much more disappointing results with essentially almost no patients responding whatsoever. And so moving forward from this trial, what we saw was patients on steroids excluded from almost all subsequent studies. In terms of PD-1, our first trial was with pembrolizumab, a trial that included both melanoma and lung cancer patients and showed that in melanoma patients with brain metastases, an intracranial response rate of about 20%. And I should point out that all of those responses have been very durable. So an important proof of principle. However, that response rate of 20% is about half the response rate we see in patients without brain metastases. So proof of principle that immunotherapy can work in patients with brain metastases and can work durably, but not as often as we'd like or really what we need. What was really transformative were recent clinical trials with combination immunotherapy with ipilimumab and nivolumab, perhaps most notably the Checkmate 204 study led by Hussein Tabi, my colleague here in melanoma medical oncology. The Checkmate 204 study included 94 patients with asymptomatic brain metastases and showed that 55% of those patients achieved an intracranial shrinkage of the tumor, an intracranial response, and importantly, over 90% of those responses are still ongoing. And so overnight, this became sort of a new treatment option for patients with asymptomatic brain metastases. And notably, it did still have significant toxicity like we see in other patients, but we didn't see any new or specifically unexpected neurologic toxicities, reinforcing the safety of this approach. And the second trial done by Australian investigators had very similar results. Now, it's not published yet, but Dr. Tabi did report at ASCO in 2019 testing of a small cohort of patients who did have symptoms due to their tumors, including some patients who were on low-dose steroids. And in those patients, we actually saw a much lower response rate. So still a treatment that, while very good for patients with asymptomatic brain metastases, really doesn't address those patients who have symptomatic tumors, which is really not uncommon. Now, I mentioned that we also have multiple targeted therapies for patients with BRAF V600 mutations present in about 50% of patients. And here we move from single-agent BRAF inhibitors to BRAF-MET combinations based on improvements in progression-free survival, overall survival, and response rates, and actually better toxicity profiles. The only BRAF-MET combination that's been evaluated in a large number of patients with BRAF mutant brain metastases is dabrafenib trametinib. I actually led the COMBI-MB phase 2 study. We did not randomize versus another treatment. All patients got treated with this combination, but we put patients in different bins based on their clinical characteristics. The largest group is the waterfall plot you see here. Cohort A are patients with a BRAF V600E mutation without any prior treatments directed to the brain. And here again, I mentioned with chemotherapy, we had a response rate in the brain of less than 5%. And here with targeted therapy, a response rate of almost 60%. And importantly, almost 80% of patients at least had stabilization of disease. And again, no new or unexpected toxicities. And while we were very excited about those results, unfortunately, what we saw was that the majority of these responses were very short-lived. And on average, the duration of response in the brain was about 6 months, which is about half the duration that we see in patients with extracranial metastases. And indeed, in this trial, 50% of patients progressed in the brain while their extracranial disease was well controlled, and really raising questions about why are we seeing this difference in activity between brain mets and extracranial mets. And there are trials going on asking, is this simply about we need to use higher doses or drugs that penetrate the blood-brain barrier? But the other possibility is that we and others have shown that brain metastases can have unique molecular and immune features which may be driving this difference in outcomes. And so with that, let's transition to talking about the molecular biology and immunology of brain metastases. As a clinician, one of the things we really would like to do is to be able to identify those patients who are really at highest risk of brain metastasis. This could, again, impact how we monitor these patients in terms of CNS imaging, but also could identify patients that we could try to intervene on specifically to prevent the development of brain metastasis. And so this was work led by Dr. Lauren Haydu, who runs our melanoma database here, and she actually previously held a similar position at the Melanoma Institute of Australia, which is the other largest melanoma center in the world. And so in large part due to Lauren, we were able to perform a unique study combining the experience at MD Anderson and the Melanoma Institute of Australia. So this study included almost 2,000 patients who had been diagnosed with stage 3 melanoma, where melanoma had spread to regional metastases. We did surgery to take out those tumors, and now basically we're trying to understand who are the patients who are at risk of having the disease come back, and particularly come back in the brain. And overall, the incidence of brain metastasis in this group was about 17%, and specifically of patients who developed any distant metastasis, almost half of them developed CNS involvement at some point in their disease course. And in terms of the clinical factors that we saw that correlated with increased risk of brain metastasis, we did see some traditional prognostic factors in this disease. We know that men have worse outcomes than women. Interestingly, earlier age correlated with increased risk of brain metastasis. And then consistent with the work that we've done before, the higher the AJCC stage or risk group, the higher the risk of brain metastasis was, albeit with some difference in kinetics. There were, though, two factors that aren't incorporated into our traditional models, including the AJCC, that correlated with increased risk of brain metastasis, which were quite interesting. And first was where the patient had their primary melanoma. And so what's shown here is over time, what percentage of patients develop brain metastasis based on where their primary tumor was. And this outlier line shown in green are patients whose melanoma was located on the scalp. And again, this is a disease where we see metastasis as tumors traveling through the blood. It's not a direct invasion, but again, a very striking association that patients who have melanoma on their scalp have a much higher risk of developing brain metastasis. The other feature was another feature of the primary tumors, and this was primary tumor mitotic rate. We actually used to include mitotic rate in the AJCC models for patients with very early stage melanoma, but in our most recent iteration, it's not part of the model anymore, but we know that it's been associated with increased risk of metastasis. What we really observed here was a very dose-dependent relationship with increasing number of metastases correlating with higher and higher risk of developing brain metastasis. And interestingly, we looked at these patients not only at their initial diagnosis of stage 3 disease, but also looked again for patients who are free of disease after 1 year, after 3 years, after 5 years. And the mitotic rate at every single time point we looked at remained associated with risk of brain metastasis. And so just on the clinical side, this really has sort of triggered this idea that these patients with primary tumors on the scalp with high mitotic rate may be the patients where we need to watch the CNS most closely. Now again, those are clinical features. There is evidence in multiple diseases that there can also be molecular features of the primary tumors that predict brain metastasis. In breast cancer, we see the highest risk in HER2 positive and triple negative patients, and in lung cancer there seems to be a correlation with those oncogenic driver mutations. And in melanoma, we had shown previously a higher risk of brain metastasis in patients not only with driver mutations in BRAF, but the second most common driver mutation in the MAP kinase pathway with NRAS. And then this very interesting association I'm showing here on the right, which is loss of the PTEN tumor suppressor. And to me, this was quite interesting because, again, I initially did my MD PhD work on the PTEN tumor suppressor, which was discovered as an event that was frequently lost in glioblastoma. So something that we had implicated in primary brain tumors. Unexpectedly, what we showed was that among BRAF mutant patients who had loss of PTEN, 100% of those patients had developed CNS disease within 2 years of surgery for stage 3. Can't be the only predictor because, again, patients with PTEN protein present also had a high risk of brain metastasis. But again, a very striking association that we saw with loss of PTEN that was validated in animal models as well. And importantly, combining both our previous molecular work and this clinical work, we now have a multi-investigator grant from the American Cancer Society and Melanoma Research Alliance to now integrate clinical, molecular, and immune risk factors in trying to identify the predictors of brain metastasis in our melanoma patients. And as we now move into an era in which those therapies that were approved for stage 4 are now approved in the adjuvant setting, to ask whether these same predictors hold up in the setting of patients receiving adjuvant therapy. Now, again, in terms of the molecular features of brain metastasis, one of the things that can drive their unique features are features that cause patients to develop brain mets, but there's also data that tumors that make it to the brain may be significantly different compared to tumors in other sites, potentially through interactions with the tumor microenvironment. And so one of the studies that we had done several years ago was looking at patients who were treated here at MD Anderson with metastatic melanoma who had undergone surgery to remove one or more brain metastases, who had also had other metastases resected during their care. And at that time, our technology was very limited. We had to use frozen tissue, so it was a relatively small study. But when we compared the brain mets and the extracranial mets from patients with both samples, we saw that the driver mutations looked really quite identical. And at the RNA level, it was a small cohort, didn't really see striking differences. But when we did proteomic analysis using reverse phase protein arrays, the striking association we saw was that in every single patient, the brain mets had higher activation of the PI3 kinase AKT pathway, which is again what's activated by loss of PTEN. However, we saw this even in the absence of PTEN loss. But again, a very interesting corollary, again, not only to the loss of PTEN in stage 3, but again to that earlier work on glioblastoma. And similar results were shown independently by a group in Germany. And to be honest, we really hadn't worked out the mechanism of this, but some very elegant work by DYU's group showed that in melanoma models and in breast cancer models that when tumor cells travel to the brain, they can actually pick up microRNAs delivered from exosomes from the tumor microenvironment in the brain that can downregulate PTEN expression. Again, we didn't always see PTEN expression in the brain, but it is this sort of interesting convergence of multiple different investigations on the PI3 kinase pathway as being important in brain tumors. Now again, almost all of these approaches were relatively focused, and I think though it did highlight the need for broader and particularly global characterization of brain metastases as those technologies became available. And really sort of supporting that, one of the most interesting studies in the field of brain metastasis was done by Priscilla Brastianos in 2015, who we invited down here for for cancer medicine grand rounds. It seems like a long time ago, but I think it was about a year and a half ago. And Priscilla had done a study where she performed whole exome sequencing on 104 brain metastases that had either primary tumor and occasionally extracranial metastases from individual patients across a wide variety of cancers. And what she was able to show was that in 50% of the patients, they were able to detect mutations in the brain metastases that they hadn't been able to detect in the primary tumors. Further digging showed that those mutations were there in the primaries, but they were there in rare subclones that weren't really picked out unless you looked for them very carefully. But these included now new oncogenic events that could be detected in the CDK4 pathway, in the PI3 kinase AKT pathway, all quite targetable. And Priscilla is actually leading a national clinical trial that's now open here at MD Anderson, where patients with brain metastases can have a brain met resected and sequenced. And if we find an alteration there that's targetable, a multi-arm clinical trial will actually allow them to go on to treatments targeting the new alterations that were seen in the brain metastases. So again, a very important study. However, from my perspective as a medical oncologist, it still left a couple things unclear. This primarily was comparing primary tumors to brain metastases, and while that's important as a medical oncologist, when I see a patient in clinic who has brain metastasis, their primary tumor usually has been removed years and years ago. What I really need to know is are there differences between their brain met and their lung met and their liver met and their bone met? And so there really was a need to compare brain mets to other types of metastases. And the other thing is, again, is whole exome sequencing incredibly important and powerful, but it really is hardwired events. It doesn't pick up the epigenetic type changes that could be generated due to interactions with the tumor microenvironment. And so with that, we conducted a study, and this was led by Grant Fisher, who's shown here on this slide, who was a very talented MD PhD student who's now a pathology resident up at Harvard. And so Grant, working with several of our pathologists, went back through all of our tissue holding for resected brain metastases from patients with metastatic melanoma. We reviewed them to make sure, again, that the tissue quality would be sufficient for DNA and RNA isolation. And importantly, we were using FFPE tissues due to, again, multiple technical advances that have been made. And in the end, Grant was able to successfully isolate RNA from 88 melanoma brain metastases from 76 patients, so a handful of patients with multiple brain metastases. We then went through, again, largely with the help of Lauren Haydu, to identify extracranial metastases that had also been resected from those patients as part of their care, and were able to identify 50 extracranial metastases from 34 of these patients. And again, to really get a global picture of what goes on in brain metastases, we prioritized analysis by RNA sequencing to get a global picture of the features of these tumors. We did do whole exome sequencing on a subset of the matched pairs and also some protein-based studies, primarily for confirmation. And so moving forward, this is the largest collection of brain metastases that have undergone whole exome, have undergone RNA sequencing ever. And so one of the first sort of questions was how heterogeneous were these tumors? And when we clustered them, they really broke up into clusters driven by the degree of immune infiltration of these tumors. And for those of you who don't follow the melanoma field very closely, in every single study that we've ever done looking at RNA sequencing data, the driver of clustering almost always is the degree of immune infiltration. So that same heterogeneity that we see in primary tumors and regional tumors, we see in brain metastases as well. And we did see that the patients who had multiple brain mets, they tended to cluster together. And one of the important correlations of this is I showed that patients who had a higher degree of immune infiltration in their tumors had better survival than patients with lower immune infiltration. And notably, this is again, primarily patients who underwent surgery before the development of our contemporary immune and targeted therapies. So really about sort of the natural history of these tumors. Now, with patients with high immune infiltrates doing better, we're very interested to understand what is it that regulates that immune infiltration. And so we looked at the molecular features of these tumors. There was no correlation with things like total mutation burden or specific oncogenic mutations. Interestingly, the one correlation that we saw with the degree of immune infiltration was that tumors that had previously been radiated had more immune infiltration than non-irradiated tumors. And this actually sort of potentially fits and maybe even explains observations that have been made by many investigators looking back at patients with brain metastases who've received radiation, where we seem to see better outcomes in patients who've gotten radiation followed by immunotherapy as opposed to radiation alone. And this is leading to prospective trials, but as far as I know, this was the first sort of direct evidence from patients supporting the rationale for those types of studies. Again, the unique opportunity that we really had, though, here was comparing the brain mets and extracranial mets from individual patients. And so when we threw all the samples in together and did clustering, they really clustered by patient, not by metastatic site. So we didn't see a true global difference between the brain mets and the extracranial mets that showed up very easily when we used this sort of universal approach. However, we did see about 500 genes that were significantly different between the brain mets and the extracranial mets. And so Grant used different pathway type programs to ask what pathways were really enriched or decreased in the brain metastases compared to the extracranial metastases. And really, no matter what approach he used, what was the consistent thing that we saw was that the brain metastases had evidence of decreased immune infiltration compared to the extracranial metastases. So even though the immune infiltrates among the brain metastases were heterogeneous, consistently they were less than what we would see in the extracranial tumors. And we validated this by immunohistochemistry. For those of you who are interested, when we looked at PD-L1, we saw that the PD-L1 status was concordant in about 60% of tumors. It was discordant in 40%, but it was actually a mix in which way that discordance was. There wasn't sort of a consistent difference one way or the other. In terms of pathways that were enriched in brain metastases, we did see a couple of gene sets pop up that were related to neuronal differentiation. And this to us was very interesting. Josh Fidler and Yusong Lee had previously shown in animal models that when they implanted tumors in the brains of mice, there was evidence that they underwent neuronal differentiation. And so that would be sort of, again, a very nice sort of validation of those preclinical studies. But we also recognized that this signal that we could be seeing could be due to the contamination by normal neuronal tissue in the samples. And so to try to really ask what was happening in the tumor cells in the brain, Grant did an experiment where he took 4 different human melanoma cell lines, injected them into the brains of nude mice and subcutaneously harvested those tumors, did RNA-seq, and then used the genome filter to really be able to isolate specifically the human RNA, which would be coming from the tumor cells, as opposed to the murine RNA that would be coming from the microenvironment. And so with that, asked what was really different in the RNA in the brain metastases versus the sub-Q metastases. And the first finding was that we did not find any enrichment of neuronal genes in the melanoma brain metastases. And I have to say, Grant and I myself were actually quite disappointed. We thought that that had been sort of a really intriguing observation. But what Grant did see was a very striking correlation between brain metastases and enrichment of metabolic pathways, and in particular, enrichment of oxidative phosphorylation. So the table below shows the pathways that were significantly enriched in the brain metastases. We were quite interested to see that not only was OXPHOS enriched, but mTORC1 signaling, because Bashananda in our lab, as I'll describe in a little bit, had previously shown that in melanoma, we frequently see tumors developing resistance to targeted therapy due to upregulation of OXPHOS that was actually regulated by mTOR. So this was biologically consistent. Also know that MYC can regulate oxidative phosphorylation. And it was an interesting observation to us, not only because, again, of our own work on oxidative phosphorylation, showing it played a role in resistance to MAP kinase pathway inhibitors, but also data implicating OXPHOS resistance to immunotherapy. So potentially a sort of central pathway that could really explain the differences in outcomes we can see with immunotherapy and targeted therapy in brain metastasis. Now again, whenever you're sort of analyzing this, you really want to know, well, do we see this in other systems? So one of the things that we did, again, that model was, as some have described, it's not really a brain metastasis model, but a brain tumor model, because we were doing direct injection. But Sherry Holman at the Huntsman Cancer Institute has the only spontaneous brain metastasis model we have in melanoma, tumors that have both BRAF mutations and loss of PTEN, and then additionally, when you add activation of PI3 kinase on top of that, these have a very high rate not only of developing primary tumors, but also lung mets and brain mets. And so Sherry generously shared tumors with us, and we showed that when we looked at oxidative phosphorylation, the highest levels were detected at the RNA level in the brain mets, which were higher than the lung mets, which were higher than the primary tumors. And so with that, we went back to our clinical data from the patients and now looked across all metabolic pathways. And when we did that, we again confirmed that oxidative phosphorylation was significantly upregulated in brain metastases compared to extracranial metastases, and indeed was enriched more than any other metabolic pathway. Again, our previous work, as well as work by others in melanoma, had shown that the OXPHOS phenotype is critically regulated in melanoma by a gene called PGC1 alpha. And so when we looked at PGC1 alpha expression in the individual pairs of brain mets and extracranial mets, showed that almost 70% of the patients had significant increase in PGC1 alpha in the brain mets compared to the extracranial mets. Now, again, all of this is at the level of RNA, so we did direct metabolite profiling as well. And so LCMS showed that we did detect increased levels of OXPHOS metabolites in the brain mets compared to the extracranial mets, again, using the model of the sub-Q tumors versus the intracranial tumors. And then Grant collaborated with Ralph DeBardinis up at UT Southwestern to do in vivo metabolic tracing, and showed that when we implanted tumors again in the brain or sub-Q and then infused them with labeled glucose, we didn't really see any significant differences in glycolysis between the tumors, but the brain met showed increased uptake of glucose. And so we were seeing functionally this increased utilization of oxidative phosphorylation in the brain metastases compared to the extracranial metastases. So with that, we felt very comfortable about the idea that this high OXPHOS phenotype was present in the brain metastases. The question was is it functionally important? Now, as I mentioned, Varshnanda had previously done work published initially in Cancer Research in 2014, showing that about a third of melanomas with acquired resistance to our FDA-approved BRAF and MEK inhibitors show this marked induction of OXPHOS. It really correlates with increased mTOR activity and increased PGC1 alpha. And we showed this in both cell lines and in patients. And last year, he did work with the IAX OXPHOS inhibitor 10759 and showed that when we tested this in vivo in some of these high OXPHOS models, it was not only very effective, but it was actually selectively effective in tumors with high OXPHOS. So just as an example, 8375 is a BRAF mutant human melanoma cell line with relatively low OXPHOS. It's very sensitive to a MEK inhibitor. When we treated with OXPHOS, we got tumor stasis, but no tumor regression. We then selected the 8375 with a MEK inhibitor until we got cells with acquired resistance, and these clones happen to have high OXPHOS. And so when we repeated the same experiment again in vivo, here you can see the cells are now very resistant to the MEK inhibitor, and now the OXPHOS inhibitor isn't causing tumor stasis, it's actually causing tumor regression. And again, working with David Pinnaker-Warren's group, we were able to show that we could actually monitor this non-invasively with FASA imaging. So with that, we asked whether the OXPHOS inhibitor would be effective in brain metastases, and we here specifically looked at models of BRAF mutant melanoma with either acquired or de novo resistance to BRAF inhibitors. And again, to be honest, we weren't expecting the OXPHOS inhibitor would work by itself. We thought we'd have to combine it with other things, but in both models, and particularly the SK-MEL5, the OXPHOS inhibitor as a single agent significantly improved the survival of mice with these intracranial tumors of high OXPHOS melanomas, even curing about 30% of the SK-MEL5 mice. We then went back to Sherry Holman and asked if we could treat her mouse model, and so she induced primary tumors. Then we started treatment with the OXPHOS inhibitor, and there was really no impact whatsoever on the primary tumors growth. And really in these mice, they ended up having to be sacrificed because they got very large primary tumors, and so we saw no difference in survival. But when the mice were sacrificed, we harvested all the tissues and then analyzed them for metastases. So what we can see here in terms of the incidence of lung metastases, mice treated with vehicle versus the OXPHOS inhibitor, a slight but non-significant difference. But what was striking was that in terms of brain metastasis, treatment with the IX drug, we saw no brain metastases whatsoever in any of the mice. And again, as I mentioned, we've often had clinical trials that excluded patients with brain metastasis, and this finding really suggested that maybe targeting OXPHOS is a specific or preferential therapeutic target in brain metastases, i.e., maybe that's the population you really have to test these agents. And so this is some unpublished data that we've generated. We're sort of wrapping up in terms of heading towards a submission. But again, one of the early groups had worked again on PGC1 alpha and melanoma. The Puig server group had taken the A375 and a second melanoma cell line and done stable knockdown of PGC1 alpha and generously shared those lines, and again, does a nice job in inhibiting OXPHOS. When we implanted those cells sub-Q versus a scrambled shRNA, no significant difference in the growth of sub-Q tumors, but when we implanted those cells intracranially, knockdown of PGC1 alpha resulted in a significant improvement of survival. So again, this idea that we were seeing better efficacy in cells implanted in the brain, despite the fact that's, again, where usually treatments are less effective. So again, really sort of important therapeutic implications in terms of where OXPHOS is important and how we should think about developing OXPHOS inhibitors, and this idea about having trials not only that don't exclude patients with brain metastases, but maybe that are specifically for patients with brain metastases. The other really interesting and clinically important observations was work where we've collaborated with Jason Hughes, whose lab is right next door. Jason is a very impressive and very productive physician scientist in neuropathology. And so Jason had similarly collected brain metastases from patients with cancer here at MD Anderson with matched primary tumors or metastatic disease, including breast cancer, lung cancer, and renal cell cancer. And in every single tumor type, the two sort of striking observations in the brain metastases was a decrease in immune infiltrates and an increase in oxidative phosphorylation. So this discovery that we made in melanoma looks like it may actually be global across different types of brain metastases. And to me, again, really suggests this may be really driven by the impact of the tumor microenvironment. And also showed in models, including here some work with Dr. Debab, again in translational molecular pathology, a breast cancer brain metastasis model, how treatment with the OXPHOS inhibitor really prevented or eliminated the development of brain metastases. This work is under review, and I thank Jason for sharing this data for today's presentation. Now, again, we saw really great results with the IX-OXPHOS inhibitor. Unfortunately, when we tested that OXPHOS inhibitor in patients, we ended up having to stop clinical development of that agent due to toxicities. And so I think we now know that OXPHOS is important to the brain tumors, and the question is how can we safely target it? And so since we have both our high OXPHOS cell lines and we again have our brain metastasis models, we've been utilizing these to evaluate different actionable strategies. And so as an example, one of the strategies that was really sort of implicated by our work was targeting glutaminase. So there had previously been data shown by others who had similarly seen this induction of a high OXPHOS phenotype in melanomas with acquired resistance to BRAF inhibitors that showed that cells switched from having a dependence primarily on glucose to really actually primarily using glutamine as their energy source and carbon source. And we similarly saw that increase in glutaminase metabolism in our acquired resistant cell lines. And when we looked at our data from patients, we similarly saw an upregulation of glutamine-dependent pathways in brain metastases compared to extracranial metastases. And while in general it's felt that glutaminase inhibitors don't really have much single-agent activity, here are again a model of acquired resistance and de novo resistance to BRAF inhibitors, where we can see when we inject these cells intracranially and treat with CD839, a glutaminase inhibitor that was very well tolerated in patients, significant improvements in overall survival in both of these models. And so we now have several members of our lab, again, using models like this to look at actionable strategies that we know are safe in patients to ask, will they inhibit OXPHOS and be effective in brain metastasis, including glutaminase inhibitors, including biguanides, and also based on some exciting work by Michael Andreeff, agents that block the mitochondrial clip or induce the mitochondrial clip-P complex. Finally, just coming back, again, I mentioned that we had worked primarily on targeted therapy resistance, but OXPHOS has been implicated in resistance to immunotherapy, both by Greg Delgoff at UPMC and then also by Mike Curran here at MD Anderson. We went back to our brain metastases and actually stratified them into those with the highest versus lowest levels of OXPHOS and looked for immune correlates of OXPHOS in these tumors and showed that, again, the tumors with high OXPHOS did have lower immune scores, which sort of has a global reflection of the immune status of these tumors. But actually, when we did CyberSort and looked at immunohistochemistry, we didn't see any difference in the number of cells that were infiltrating those tumors. And this is very consistent with work by Dr. Delgoff that really implicated OXPHOS in the activation status of the immune cells in tumors more than the degree of immune infiltration. And we similarly see these signatures of decreased immune activity in the high OXPHOS brain mets versus the ones with lower OXPHOS. And metformin in our models can induce the expression of many genes related to immune cell activation and has shown at least an additive effect when we combine it with immunotherapy in our brain met models. So just to then summarize, again, it's really been exciting to be a part of this work and a part of the team that we have not only here at MD Anderson, but really across the oncology field focused on brain metastasis. And so, again, for those of you who are interested in the clinical realm, clearly a lot of progress on the therapeutic side and really have established combination immunotherapy with ipilimumab and nivolumab as a real viable option for patients who have asymptomatic brain metastasis. And that being said, our work's not done. 50% of these patients have no response at all. This regimen does have toxicity, and the question is can we do better? For targeted therapy, our clear challenge is how do we make the responses more durable? And what we're doing in the clinic is really moving to combinatorial approaches. And one of the things that really facilitates those types of investigations here at MD Anderson has been the development of the MD Anderson Brain Metastasis Clinic, co-led by Dr. Tabi from Medical Oncology, Dr. Fred Lang from Neurosurgery, and Dr. Jing Li from Radiation Oncology, a place that's really allowing us to facilitate not only high-quality care, but high-quality research for patients with brain metastasis, regardless of what tumor type that they have. And so again, then coming back to the laboratory, the key research findings that we're sort of seeing is this growing evidence of unique features of brain metastases, novel mutations and differences in both oncogenic pathways, immune infiltrates, and metabolism. And again, I think the really intriguing finding that some of these phenotypes at least seem to be shared across cancer types, again, suggesting there may be unique sort of requirements for a tumor to grow in the brain and therefore potentially unique vulnerabilities. We do know that many of these features that we're seeing in brain metastases are associated with resistance to both targeted immune therapies, and so some very highly probably translational type concepts. And I think these insights also suggest some rational combinatorial strategies, not just our OXPHOS stuff, but PI3 kinase pathway and even radiation as a strategy to improve immunotherapy. But I think as a laboratory researcher, one of the take-home messages is this idea is I think the findings overall support the need for continued focused research on CNS disease, and one could argue really looking at all of these different organs that are really problematic for us in the clinic to ask, are there things about specifically that microenvironment that make it so challenging? The last thing I'd like to highlight is, as I mentioned, the final frontier of CNS disease is leptomeningeal disease. This is something where, again, we see average survival measured in the matter of a couple weeks as opposed to months. And we're very proud of the fact that we have Dr. Isabella Glitza leading sort of, I think, the world's best intrathecal immunotherapy program, and Dr. Charisse Ferguson from Neurosurgery, who's developed multiple new models of syngeneic melanoma models of leptomeningeal disease that we're now using to evaluate different combinatorial strategies. And again, this real partnership that we have with Dr. Glitza and Dr. Ferguson to really make that gap between our discoveries in the lab and the clinic as small as possible. So with that, again, this is something that has involved many different collaborators, not just in my lab, not just in our department, but across the institution and indeed across the world, and support from multiple funding agencies. So with that, I really appreciate the opportunity to share this sort of work this morning, and I'd be happy to take any questions you may have.
Thank you, Michael. This was wonderful presentation and wonderful data. Congratulations, excellent clinical and translation research. And in fact, the attendees said beautiful work, Michael. And let's start with the question, because these are the most important. So regarding the brain metastases from scalp lesions, are those metastases parenchymal or meningeal? And then, are these anatomical connection between scalp circulation or meningeal dural?
It's a really great question. It's one of the things that my assumption is that it's really primarily with parenchymal brain metastasis. We've actually expanded that study that I described with stage 3 patients recently to now collect data on several thousand, about 5,000 patients diagnosed with primary melanoma, stage 1 and stage 2, and have collected the data about the incidence of brain metastasis and leptomeningeal disease. You know, what's interesting is when I've looked under the slide at many of these models that we've developed in patients with LMD, it looks like very often what LMD represents is the parenchymal tumor having the ability to migrate and invade out to the surface of the tumor. We do sometimes see patients with LMD only, and so again, it's not that you can't have both types of things, but to this point, primarily seems to be parenchymal brain metastasis. But I would say that what you've asked is a really good question. I don't think we've specifically asked that about the patients with scalp primary. So I thank you for the question. It's a really good thing for us to go back and look at.
Now the continuation of the question, do you have insights on the genomic determinants of resistance to targeted and immune therapy? What is the role of BRAF dosage on resistance?
Yeah, so a really great question. So this is a place where, again, in terms of genomic determinants, you know, in the BRAF space, one of the interesting things is that we haven't seen the loss of the BRAF mutation, and we have not seen mutations in the coding region. We do see about 20% of patients at the time of resistance with amplification of BRAF. When that happens, you can overcome it just by increasing the dose of BRAF, at least in preclinical models. And we do, interestingly, see alternative splicing of BRAF to get a smaller protein. And for that, no matter how much BRAF you add, you won't overcome it. But they're still driving the MAP kinase pathway. MEK inhibitors should block that. In terms of other mutations, about 20% of patients at the time of progression now have a concurrent NRAS mutation to complement their BRAF mutation, something we don't see in patients who are treatment naive, but something we see in about 20% of patients that have progressed. So those are really the main genetic drivers that we see. It's interesting, about 40% of patients, it all appears to be epigenetic. And so it's a real mix of genetic and epigenetic mechanisms. And similarly, you know, resistance to immunotherapy, there are some things that we do see that are genetically driven. It seems like the JAK2 mutations, loss of beta-2 microglobulin, and loss of, again, presentation of antigens are certainly seen. We've seen that loss of PTEN can cause resistance to immunotherapy and have trials combining PI3 kinase inhibitors with PD-1. You know, again, I think that we know that there's multiple different mechanisms of resistance to immunotherapy, whether it's about trafficking of T cells into the tumors versus being excluded, or situations where you can inactivate the T cells, or where the tumors just have intrinsic resistance. Again, still a field where there's not really a consensus on exactly what the mechanisms are. There seem to be many different mechanisms, and I would point out one of the things we really don't understand is when patients have multiple tumors progressing, are they progressing because of the same mechanism or different mechanisms? We know in targeted therapy it can be very different mechanisms from one tumor to the next, or even from one region of a progressing tumor to another with the targeted therapies, but we haven't really figured that out well for the immunotherapy yet. So immunotherapy resistance remains very complicated, with many different types of factors contributing, not one consistent genomically driven mechanism of resistance.
Thank you very much. So I continue the question, and only one point here. Basically, I thanks to all the people who are questioning. We were advised not to say the name of the people who is asking. So it's not that I don't like to say your names, but basically we have to keep some rule because this is recorded presentation. And let's go to the next question. Thyroid cancer also has similar mutation, including BRAF, P53, and so on. Any thought on thyroid cancer?
Well, in thyroid cancer, again, with BRAF mutations, we do know that the BRAF inhibitors can be very effective. We see this not only in thyroid cancer, but also in lung cancer. There's certain types of primary brain tumors. And it does seem, and actually one of the first examples was colorectal cancer. There's a subset of tumors that have BRAF mutations. And what's interesting is that in all of these tumor types, BRAF inhibitors have some activity, but it's much higher in some cancers than in others. So a very good friend of mine, Scott Kopetz, and I came through fellowship together. He was working on, again, molecularly targeted approaches for colorectal cancer and started working on BRAF mutant colorectal, and showed that while in melanoma we had response rates of 50% with BRAF inhibitors, in colorectal cancer it was about 10%. And Scott very elegantly showed that it was because in colorectal cancer you also had to target the receptor tyrosine kinases that were expressed in colon cancer that weren't being expressed in melanoma, leading to Scott really driving the development and recent FDA approval combining EGFR inhibitors with BRAF inhibitors. And so there are examples. My recollection in thyroid cancer, Maria Cabanillas would be the person to ask. I think the efficacy of BRAF and BRAF-MEK in thyroid is pretty similar to what we see in melanoma. And certainly this is a place where there's the overlap with P53, there's the overlap with PTEN loss as well. There's many diseases that sort of have these shared pathways, and hopefully we can again extend lessons learned in one cancer to other cancers as well.
Thank you. Another interesting question. Would be advisable to switch from targeted therapy to immunotherapy prior to developing resistance to targeted therapy?
It's a really great question, and we face this on a daily basis in melanoma. It's again, melanoma and lung cancer, again, are these two diseases that are really being transformed by both immune therapy and targeted therapy. What's so interesting is in lung cancer, you have the population of patients that have those targetable driver mutations, EGFR and ALK, tend to happen in patients who have a very low mutation burden. And high mutation burden is very important in lung cancer for responsiveness to PD-1. So they're really almost separate populations. In melanoma, we actually have complete overlap. The BRAF mutations are present in tumors that have very high mutation burden, and we know that immunotherapy and targeted therapy both work in those patients. We have wondered in these patients, there now are still clinical trials ongoing to test for a BRAF mutant patient, should you start with targeted therapy or should you start with immunotherapy? And then as I mentioned, this year we had the first approval of a BRAF inhibitor plus MEK inhibitor plus PD-L1 inhibitor. Should you really give them together? But this question of how should you really sort of, again, sequence or switch, it's a really great question. There's also data that the targeted therapies, when we block the MAP kinase pathway, and this was really an insight that Jen Wargo made very early on when she was working at Mass General, is that targeting the MAP kinase pathway in melanoma not only slows the growth of these cells, but it makes the cells more recognizable to the immune system. So there's a very strong rationale for using targeted therapy as a way to increase your responsiveness to immunotherapy. But we're still waiting for really good clinical proof to really tell us how we should be going back and forth or combining these together.
Great. I will take a couple more questions. So glutaminase inhibition also reduce glutamate and consequently glutathione. Does increase in cytoplasmic growth and not OXPHOS influence metastatic cell survival in high glucose central nervous system environment?
It's a really good question, and I have to say we're probably now getting to questions that I know that we don't have the answer to. I would highly recommend anybody who's interested in talking about this, one, could reach out to me, but also Vashish Nanda in my lab has a paper under revision looking in greater detail at glutaminase inhibitors in melanoma. But again, certainly we do not have all of those answers worked out. I would argue our data with the glutaminase inhibitor in brain mets is extremely preliminary. It's really pilot experiments. But I think the results and the fact that we know that the glutaminase inhibitors are so well tolerated makes us think that this is a strategy that that really deserves more study, both preclinically and clinically. And again, would be really appreciate the chance to talk to people with ideas about questions like the one you're asking.
One more question. Have any agents that preferentially inhibit slow-growing cancer cells been tried as inhibitors of brain metastases?
It's a really good question. I mean, I think that, there's not a great understanding at this point. And again, if somebody has data, please correct me. With immunotherapy, I don't get the impression that we really think there's sort of a cell cycle dependency to the efficacy of immunotherapy. The way that immune cells will recognize and attack cancer cells isn't really sort of, again, dependent on inducing DNA damage. And so if you sort of think about immunotherapy being something that doesn't really seem to, again, be dependent on whether the cells are fast or slow growing, I think that's one that you could sort of ask that question. And clearly there is efficacy for immunotherapy in brain metastasis. In terms of the slow-growing cells, again, I don't know. I think one of the questions is what would you specifically say are examples of those types of therapies? But I don't think it's been very specifically. One thing I will say is that Maynard Herlin defined a subset of tumor cells within melanoma, slow-cycling cells with expression of Jared1B, as potentially, again, sort of the cells that are able to drive therapeutic resistance when you treat them with different therapies. And he actually showed that one of the distinct features of those cells is that they have high oxidative phosphorylation. They don't actually seem to be driven by PGC1 alpha, but they do seem to, again, have this OXPHOS phenotype.
Thank you. Now, the last one, a continuation of the question on targeted therapy, immunotherapy. The continuation is, but those who develop resistance to targeted therapy, OXPHOS upregulation, have resistance to immunotherapy. So, should we shorten the time of targeted therapy?
Yeah. Well, I mean, what we sort of know is, again, and again, this debate about should you start with targeted therapy or immune therapy, we actually know that for patients who are BRAF mutant, if we start them on targeted therapy and they progress, their responsiveness to immunotherapy goes down. If we start BRAF mutant patients on immunotherapy and they progress and we treat them with targeted therapy, we also know that their responsiveness to targeted therapy goes down. In each of these cases, we're sort of ending up with a bad version of cancer that seems to be more resistant to the other therapeutic approach. But again, in terms of what you're suggesting, there is data, again, that targeted therapy resistance can be driven by upregulation of OXPHOS, and when you have, again, activation of the MAP kinase pathway, you suppress the expression of melanocytic antigens, all of things which make immunotherapy tougher to really sort of work. And it really does drive, as you mentioned, even when targeted therapy is working, the question, should we switch patients to immunotherapy? But it's also the interesting observation, patients who don't respond to immunotherapy also have shorter responses to targeted therapy. So I think, again, theoretically, you're bringing up a good point. We just, again, it's one of those things that still remains a challenge in terms of exactly how to sequence these drugs.
Thank you. And last question, let me to ask you one question because I want it from long time. So when you get brain metastasis versus non-brain metastasis, you didn't find gene expression differences too much. But did you ever look to the non-coding part of the DNA? So whole genome sequences, enhancers, super enhancers, ultra-cancer region, these sides of the sequences that not too many people are doing these days.
Yeah, I mean, so we did whole exome sequencing. We did not do whole genome sequencing. So we probably haven't answered that question. We've done some exploratory studies looking at microRNAs, and actually, with you asking the question, we should probably sit down with you and share the data that we have. But I think, George, it's a really good question that I don't think anybody has an answer for yet.
Thank you. So with this, Michael, thank you very much. Really wonderful presentation and a lot of questions. So the people were really very interested. Here in 2 weeks from now, Lauren Byers, another wonderful clinician scientist with an excellent presentation. And I want to tell thank you all for being here with us. keep using mask, because mask will not stop us to make discoveries or treat well the patient, will stop widespreading the virus. And with this, waiting for you in 2 weeks from now. Thank you again, attendees, Michael, and the people who organize, Liz and Laura, Adame, and see you in 2 weeks. Good luck to all. Thank you for being with you. Thank you, Michael.
Thanks, George.